what is the neural development, and what is the primary structure that is very important that needs to remember is the how is neural tube formed and how is the neural tube is formed. This process of formation of neural tube and a neural crest cells is called neurulation. So first of all, you should understand the concept of neurulation. Okay, so the primary neurulation process is that, okay, how this neural tube and a neural crest cells are formed. So I will go and like let you show a picture how this neurulation is happening. Okay, so in this diagram you can see the concept of neurulation, that first of all you should know that there is a development of the three important structure. Okay, the first structure is very important, is ectoderm. Okay, and next is very important, that is the mesoderm, and third structure is very important, that is the endoderm. So these are the three structures that you should know about. And what is this? What is this? Our importance is ectoderm. Ectoderm is the layer that is very important for the neurulation. So this ectoderm is now very important for our topic. This ectoderm will make all the CNS system, all the structures. Okay. Now we are discussing neurulation process. Neurulation process. What is the process of neurulation? The formation of neural tube and the neural crest cells. Okay, so first of all, you can see in this diagram there is ectoderm here, and in between this ectoderm and this green color, which is endoderm, I will highlighting with green color, this is endoderm, and in between them this is structure is mesoderm. So ectoderm, mesoderm, endoderm. In between ectoderm and endoderm there is a mesenchymal structure, and in between these two mesenchymal structure there is a very important yellow color notochord. Okay, so this is notochord, this structure. Okay, the first importance is notochord is that this induces, the keyword of MCQs is that this induces the neural plate. Okay, what happens next? When notochord induces this neural plate, then what will happen? This neural groove and this neural fold is made. After further notochord induces this neural groove, neural fold, and then this structure is made. You can see this. And after that, the this ectoderm is separated from neural tube. Remember, in this concept of this blue color, this is ectoderm here. And now you can see that, now you can see in this structure that this is separating. Okay, this is separating, and this separated structure like this here is neural tube. Okay, the first started with here that notochord induces neural plate to form a neural groove, and neural groove as well as a neural fold is made like this structure. After this neural groove and neural fold, next is a formation of neural tube. Above neural tube, this neural tube, there are cells that are neural crest cells. Is that clear? So these are neural crest cells like this. Okay, so in simple word, in simple, you should know about the neurulation process. That is very important. That is start from here. There is a neural plate and a neural plate borders. In between ectoderm and endoderm, between them is mesoderm. Between these mesoderm right and left, there is a yellow structure called notochord that induces and makes the neural tube and neural crest cells. This is a process of neurulation. Okay, so we have discussed the process of neurulation. Is that clear? Now we will come back to our topic. So we have discussed that what is neurulation. Next, we will read the lines. A notochord induces overlying ectoderm to differentiate into neuroectoderm, and it forms a neural plate. Is that clear? And then neural plate becomes neural tube and neural crest cells. And very important next, what is the adult derivative of this notochord? And that notochord will become nucleus pulposus of intervertebral disc in adults. Is that clear? So next is what are the neural plates? We have discussed that neural plates give rise to the two structure. One is neural tube, and the next is neural crest cells. Neural crest cells. Okay. Next we will now go to and discuss about what are the neural tubes lateral walls made. Lateral walls divided into alar and basal plates. Alars and basal plates. What are these? Now I will show a picture in which we will be discussing how these plates are made. Okay. So you can see in this picture, this is a very beautiful picture. You can see it. This is a neural tube. This is a neural tube. In this neural tube, these are above side is neural crest cells, and these neural tube and neural crest cells will have a different function. And you can see that this neural tube will go and make spinal cord portion. We know that there is anterior horn of spinal cord and posterior horn of spinal cord, which are adult structures. In anterior horn, we know that this is called ventral gray horn, and posterior is dorsal gray horn. So the neural tube will be making like in embryonic structure, which is a specific name of these are ventral plate or dorsal plate. Okay, so these are the names of the embryonic, and adult name will be dorsal and the ventral. So you should know that this neural tube will be making the dorsal and ventral, and neural crest cells will be making a specific structure that will be very important, like ganglia. Okay, neural crest cells will be making the melanocytes. This is a melanocyte structure, and neural crest cells will be making the spinal ganglion, because neural crest is making the peripheral nervous system ganglia, spinal ganglia. Okay, and in more, if we go more detail, that neural crest cells will be making ganglia in the sympathetic system also. And these ganglia of the sympathetic system are made by neural crest cells, and these sympathetic trunk are available in our gastrointestinal system. Like we know that there is a celiac ganglia and GIT, that is structure of ganglia made from neural crest cells. And there are the renal ganglia structure, supra renal gland also has the medulla portion, that is also made by the these neural crest cells modification to the making the ganglia. Okay, so you should know what are the structure and that are influenced by the neural crest cells. So now we will go back and discuss our first page. What is Eller plate and what is basal plate? So I have shown a picture that dorsal Eller or dorsal, which is very important. It is induced by a specific protein that is called bone morphogenetic proteins. Bone morphogenetic proteins. Okay. So the Eller plate, which is dorsal plate, induced by the bone morphogenetic proteins. Remember the word bone morphogenetic proteins. But the basal plate, which is called ventral, which is called anterior horn in the adult structure, that is induced by sonic hedgehog genes. So there are a specific like factors and protein that leads to induction phase, like they leads to the molecular activities and leads to the development of basal plate. So this is the process of neural development. Okay, now we will go to discuss about what are the regionalization of neural tube. Okay, we have discussed at this time what is neurulation process and what are factor that making Eller are the like basal plates, and what are the factor that induce Eller plate, that is bone morphogenetic protein, and what are the factor that induce the ventral, that is sonic hedgehog genes. Is that clear? So this Eller plate will be making dorsal horn in adult structure, and the basal plate, which is ventral, will be making the anterior horn in the adult structure. Now we will go to discuss a few of the important concepts about the what is development of our central nervous system. Development of our central nervous system is via the very important is the three primary vesicles. And these three primary vesicles, the one is called forebrain, that is prosencephalon. Next is midbrain, that is mesencephalon. And the third is hindbrain, that is rhombencephalon. Okay. So forebrain, prosencephalon, midbrain, mesencephalon, hindbrain, rhombencephalon. And the forebrain, which is prosencephalon, is further divided. Okay. It is divided into telencephalon and diencephalon. Telencephalon and diencephalon. Remember, the telen will be making hemispheres and diencephalon will be making thalamus, hypothalamus, and retina portion. Okay, so this telencephalon will be making the hemispheres, and diencephalon will be making the thalamus, hypothalamus, and retina. Is that clear? And what are the important concept here? That what are the cavities in this forebrain? Okay, the cavities in the forebrain are lateral ventricle and third ventricle. Lateral ventricle, third ventricle. Lateral ventricle, third ventricle. Remember this. Lateral ventricle cavity is present in telencephalon, and third ventricle is present in diencephalon. Next is midbrain. Midbrain is mesencephalon. It is not further divided. It will go and make the midbrain. Remember, this is a midbrain, and the cavity in the midbrain is cerebral aqueduct. Cerebral aqueduct. Remember this. That is connecting the third ventricle with the fourth ventricle. That is cerebral aqueduct. Is that clear? Now, next, hindbrain. Hindbrain is rhombencephalon. Okay. Rhombencephalon. And rhombencephalon is divided into further two, and that is metencephalon and myelencephalon. Meten, myelin, meten, myelin. Remember, meten, myelin, meten, myelin. Median is further divided into two, pons portion and cerebellum portion. Pons and cerebellum, and myelin is making the medulla. Okay, how to remember this? I know very difficult to remember this, but you have to write it down on your book, on your like white pages, and remember again and again. Okay, if you remember myelin, it's contain L, so you can remember. So myelin will be medulla because medulla is also containing L. Uh, whatever is easiest for you, you can write it down on the paper and remember again and again. What is the cavity in this hindbrain rhombencephalon? That is the fourth ventricle. Remember, fourth ventricle. There is a division: upper portion of fourth ventricle and the lower portion of fourth ventricle. Upper portion is in the mesencephalon, that is making pons and cerebellum, and the lower portion of fourth ventricle present in the myelencephalon, that is making medulla. So is that clear? So we at this time we have discussed what are the neurulation and what are CNS portion development. Now we will give to discuss a few of the important image that you should know and make your concept more easier for you. Okay, so you should know about the timeline. What is the most important MCQs that can be asked in your exams? That is, what is the week the neurulation starts? Okay, at which time the development of central nervous system starts at the week 3 or starting of week 4. Okay, so the start of week 4 from the 20 like second day is the actually a starting of central nervous system development. So there are phases in fetal development timeline. You should know what is in the pre-organogenesis, that is in the first 2 week, when there is a gastrulation happening, and you know that the 3 layer will be made in the first 2 week. From the third week, there is embryonic stage. And this embryonic stage, there will be a development of different organs. Okay, development of central nervous system, you know that it is started in the third week and go till the development of central nervous system go. And most important, highlighted in the dark color, is that there is excessive teratogenic activity happen in this dark color. Remember the dark color which I am highlighting, this dark color till this, you can see that this is a dark color. It's mean there is more teratogenic activities in this area. If there is a problem like heart development happening from this the mid of third week till this 6 week, there is a more teratogenic and problem happening. So more dark color mean there is a more risk of teratogenicity. Is that clear? So you should remember, you can remember this picture. What is a timeline? What is pre-organogenesis? And what is embryonic stage? And what is the fetal stage? So now we are going to discuss central nervous system. So you should know about the central nervous system development starts from the end of the third week, start from the fourth week, from the 22nd day, 22nd of the day, and it goes till of the end. But the most important risk that happens in central nervous system is till 16 of the week. Remember this. Okay, from the third to the 16th, this is the most at risk weeks that happen leads to the teratogenic activity. So it is written here in the below section. It is a dark mean. It is highly sensitive period which having a structural anomalies. Remember, when this is having a structural anomalies, that is a dark area. Okay, so going to the next, we have discussed this picture, very beautiful picture. I am just quickly giving you a quick revision of the process of the neurulation. You should know there is the classically a picture, and you should know that there is a primitive knot, and this is a primitive streak, and this is a cloacal membrane, and this is a neural plate. And if I get this section, in this section you can see that in this is the notochord, and above this is neural plate. Neural plate has neural groove and neural fold, and then this notochord induces this neural plate and makes the neural tube along with neural crest cells. Okay, so this is a picture, very important. This is notochord here in the center. This oval is like the neural tube. This is a tube, and above this, like this, is the neural crest cells. So you should know like this one. Okay,